Hello students, welcome to BAN lectures. Today we will talk about complex lipids or compound lipids. So this video would be suitable for medical, dental, pharmacy, nursing students as well as for students studying biochemistry either at UG level or PG level. Now complex lipids or complex lipid as the word indicates itself means they are not simple. They must be containing something different from simple lipids. So in previous videos we have classified lipids into three classes: simple lipids, complex lipids, and finally derived lipids. We have defined lipids. We have discussed those triglycerides, fats, oils, etc. and waxes also. Now compound lipids, they are grouped into two classes: phospholipids and glycolipids. Now phospholipids are further grouped into glycerophospholipid and sphingophospholipid. Now glycero itself indicates the alcohol present is glycerol. Glycerol forms the backbone of glycerophospholipid, and sphingo itself indicates that the alcohol present is not glycerol but it is something different, that is sphingosine. Sphingosine forms the backbone of sphingolipids. So this is the alcohol present. Now we conclude that glycerophospholipids contain three important parts. One is glycerol forming the basic backbone. Fatty acids, they are esterified to first two carbon atoms of glycerol, nitrogenous base and phosphoric acid. Here you can see the simple structure of phospholipid. This is the basic backbone is glycerol. Here two fatty acids are esterified to first two carbons of glycerol. Next you have this phosphate group present. To this phosphate group is attached as something nitrogenous base. Now simplest phospholipid does not contain nitrogenous base. Now let us see the next slide. This is the simplest structure of certain complex lipids. Now this is glycerophospholipid. You can clearly understand this is glycerol backbone. This is fatty acid esterified to glycerol. Another fatty acid esterified to second carbon atom of glycerol. This is phosphate group attached. This is alcohol group attached. This is actually amino alcohol. Now in sphingolipids, the backbone is formed by alcohol, that is sphingosine. This is fatty acid esterified to sphingosine. Then you have the phosphate group, then you have some amino alcohol. Similarly, glycolipids, this is the sphingosine is the basic backbone. To this is esterified fatty acids, and next another group attached is a carbohydrate saccharide. It might be monosaccharide, it might be oligosaccharide, that we will see later. Now phospholipids. Now we conclude the basic structure. Of course, backbone is phosphate group, glycerol molecule and two fatties. Now what we would like to talk about is this phospholipid comprises of two portions. One is hydrophilic part, other is hydrophobic part. Now this hydrophobic part is formed by fatty acids, and the hydrophilic part is formed by glycerol and a phosphate group. Now the phosphate group present in the head of the phospholipid is hydrophilic, as we said. Now phosphate group can be of many types depending upon the type of phospholipid. It can be serine, it can be inositol, it can be choline. Now glycerol acts as a backbone in phospholipids. Now in case of fatty acids, there are long chain hydrocarbon chains. If one fatty acid is attached, then it is known as lysophospholipids, and if two fatty acid is attached, it is known as phosphoglycerides. Again you can see the structure so that you can understand things carefully. Now then phospholipid, as we just now said, hydrophilic part. Hydrophilic part is formed by phosphate present and glycerol. And the hydrophobic part is formed by fatty acids. One of the fatty acid chain is saturated, other one is unsaturated. This forms the hydrophobic part, that's hydrophobic tails. This is how phospholipids are arranged in the cell membrane. They are also called membrane lipids. This is the polar head group, and this is hydrophilic in nature, and these are the hydrophobic tails. Now glycerophospholipids may contain nitrogen or they may not contain nitrogen. So they are grouped into two classes: nitrogen containing and non-nitrogen containing. If nitrogen is present, then they might contain lecithin, and we call it phosphatidylcholine. Or it may be cephalin, we call it phosphatidylethanolamine or phosphatidylserine. Non-nitrogen containing means they might contain inositol, they might contain glycerol or diphosphatidyl They do not contain nitrogens. Now, lecithin is glycerophospholipid with choline as nitrogenous base, most abundant phospholipid of cell membrane. Now, cephalin is glycerophospholipid with ethanolamine as nitrogenous base, component of membrane, plays a role in blood coagulation. They have a very important function to play. Cardiolipin means made up of two molecules of phosphoric acid linked by a molecule of glycerol. It is the major component of inner mitochondrial membrane, only phospholipid with antigenicity. Similarly, phosphatidylserine means nitrogenous base is serine. Phosphatidylinositol means it is not a nitrogen containing group compound. It is present in cell membrane, role in cell signaling, membrane trafficking, second messenger in hormonal pathways. So you can see the structure of cephalin as we said just now. This is basic backbone is glycerol, fatty acid attached to first carbon atom, another fatty acid attached to another carbon atom of glycerol. Here you have the phosphate. This is choline, phosphatidylcholine. So this is lecithin. Here it present nitrogenous base is ethanolamine. This is inositol. We call it phosphatidyl inositol. Non-nitrogenous containing group is here as we said in the previous lecture. Sphingophospholipid, they contain sphingosine as alcohol. This forms the basic backbone. Only sphingolipid known is sphingomyelin. So sphingomyelin now contains sphingosine is attached to fatty acid by an amide linkage forming ceramide. So ceramide is linked to phosphoryl and nitrogenous base. Nitrogenous base is choline to form sphingomyelin. Sphingomyelins are present in outer leaflet of cell membrane bilayer, abundant in myelin sheath of nerve cells, play a role in cell signaling. So this is sphingomyelin. As you see, this is the sphingosine. It is the basic backbone. To this are esterified two fatty acids. Sphingosine plus fatty acids, they create ceramide. This is ceramide. To this ceramide is attached phosphate group, and then you have the choline over here. So this particular compound is sphingomyelin, only sphingolipid. Again, you can see the structure of this thing, sphingomyelin, fatty acid choline. Now, glycolipids are complex lipids. There is no glycerol present. There is no phosphate group. Alcohol present is sphingosine. To this alcohol are esterified fatty acids, and they contain a carbohydrate which might be a disaccharide like galactose, or they might contain a oligosaccharide also. So glycerophospholipids, glycerol is the backbone with carbohydrates. Sphingoglycolipids, sphingosine is the backbone with carbohydrates. Now sphingolipids can be further grouped into four subclasses: cerebrosides, sulfatides, globosides, and gangliosides. So they are complex lipids with carbohydrate, no phosphoric acid, and alcohol present is sphingosine. Now cerebrosides. Now the basic backbone is sphingosine. To this is esterified fatty acids, and sphingosine to this attached is monosaccharide glucose or galactose. Again, ganglioside, you have the sphingosine as backbone, fatty acid is esterified. To this is attached glucose or galactose, amino sugars, and nana. This is cerebroside. Again, you can see clearly sphingosine, fatty acid is esterified to it, containing carbohydrate, monosaccharide, or oligosaccharide. Sulfatides, ceramide, plus monosaccharide and sulfate, esterified. Sphingosine plus fatty acid, it means ceramide as we said in the previous slide. Globoside, sphingosine as basic backbone, fatty acid is esterified to it, and oligosaccharide is attached to ceramide. Again, ganglioside, you have the basic backbones, sphingosine, fatty acid is esterified to it, you have glucose or galactose attached, and to this glucose attached NAMA. NAMA means an acetyl new muraminic acid.